everyone, and thank you for coming. It's wonderful to see you all here on a Friday afternoon, and I heard that there was I thought city would be easy, but if you're driving, it might be a bit of a challenge. So thank you so much for finding the time in your busy schedules to come here and spend the afternoon with us. And you are in for a treat, because we have Sara here presenting her exciting research. But I would start with thanking Oliver for helping me to organise this, because in the past we've been organising these events in Melbourne University and at Monash, so it's good to be at RMIT as well. So thanks all. And Oliver is sitting over here. Already. But let's start with introduction. I'd like to introduce our today's speaker, who is Sara Long, my colleague and friend from Melbourne University. And I'm going to take my notes here so I get these correct. So Sara has a Bachelor of Science joint honours degree in biochemistry and pharmacology from King's College, from the University of London, in the UK, and a Master's of Science in Natural Research and Management from the University of Leicester. She obtained her PhD in aquatic ecology from RMIT University, so we go back at your roots here. And that was in 2001. So since then, she spent 8 years in a research institute in the UK called Mosswood, working on investigating the effects of pesticides and industrial contaminants on vertebrates and invertebrates. And I'm assuming that from there you then started at CABEM, in the Centre of Aquatic Pollution Mitigation and Management. You've been working there the last 8 years. So you're going to talk about the metabolomics, so the floor is yours.
Great, thanks Minna, and thanks everybody for coming as well. So I'm going to talk to you today about some of the work that I've been doing with a few collaborators using metabolomics to understand the interaction between aquatic organisms and their environment. So I'll just give a brief outline of what I'm going to talk about today. So I'll talk about biomonitoring briefly, then I'll introduce biomarkers and a bit about metabolomics as well. And then I'll give you a few case studies where I've worked on metabolomics approaches on freshwater, estuarine and marine environment, and then where we can go from there. So just to put where this research into context, so currently to assess water bodies we need to do a few different types of measurements. So we do physicochemical measurements, which is basically measuring the contamination in the water and the sediment, and we look at water quality parameters. We also carry out habitat assessments, so looking at the vegetation and just the general assessment of water bodies. And then in terms of biological measurements, it tends to be mainly the presence and absence of macroinvertebrates. And we're starting to introduce standard toxicity tests where we collect sediment and water from the field and bring them back to the lab and expose them to laboratory organisms and look at their responses. But at the moment there's a lack of effects of individuals, and we're looking at the effects on biological responses within an organism. And this is a bit of a downfall as well. So where we can improve our monitoring is that we can look at different levels of biological effects to assess ecosystem health. So the community level effects, it's highly ecologically relevant, and this is where the macroinvertebrate surveys come in. But there's a weak causal link to chemical exposure, so we don't know what's causing the poor condition, but we just know that the water is in poor condition. And so the individual level effects are more early response biomarkers, and these are responses with an individual or a tissue or molecular approaches. So it's low ecological relevance, but it's got a really strong causal link to chemical exposure. And so that's where this work fits into here. So there's many different definitions of biomarkers, but they all sort of say the same sort of thing, which is that they're biochemical, physiological or behavioural changes in a tissue, body fluid or whole organism that can provide evidence of exposure to chemical pollutants, and then we also indicate toxic effects. So you can have biomarkers of exposure, so they just show that animals have taken up a chemical, and you can also have biomarkers of effect to show that that chemical is having an adverse effect on an organism. And the different types of biomarkers are just listed here. So we have molecular biomarkers, that tends to be more gene expression, transcriptomics, and then we can have biochemical, physiological and behavioural biomarkers, and they just increase up the level of biological effect to looking at more whole organism responses in the behavioural effects. You can also have different specific types of biomarkers. So you can have chemical-specific biomarkers, and as the name says, they just respond to particular chemicals. So that's using biomarkers in a more diagnostic way. So an example here is metallothionein. It's a protein that's only induced or mainly induced through metal exposure. So if you see an animal that's got high levels of metallothionein in it compared to an animal from a clean site, you get a good indication that metals are present. And then you can then go on and investigate further where these metals may have come from in terms of your biomonitoring program. And there's also non-specific biomarkers of exposure. They're just general biomarkers that tell you that the animal's under some sort of stress, but you can't work out what it is. And then maybe you should go back and try and delve in a little bit deeper to find out what's actually causing the problem. And why are biomarkers useful in biomonitoring? Well, I guess the main thing is that they demonstrate uptake of a chemical. You can measure chemicals in the water and in the sediment, and they tell you what's there, but they don't actually tell you if it's bioavailable for the organism to take up. And there's a lot of new analytical techniques that you can look at bioaccessibility and bioavailability through chemical methods, but it still doesn't tell you what chemicals are actually being taken up by an animal, whereas looking at the biological response shows that the animals have taken up this chemical and it's having some sort of adverse effect. And one of our challenges is to develop new responses and new techniques to incorporate into the biomonitoring programs. So to work together with the chemical analysis and the habitat assessments and the macroinvertebrate work to design really good quality biomonitoring programs where we can start to identify what chemicals are causing the problem so we can then manage our sites better. So ideally these responses will be chemical or class specific, so we can use it in a diagnostic way. And also, ideally, they can detect changes before we see impacts at higher ecological levels. Once we start to see population declines, it's very hard to get those animals back. So if we can develop early warning markers that something's happening at a site, we can then go in early and try and improve the condition of that site before adverse effects start to happen. And this is where metabolomics comes in. So metabolomics, it's the analysis, quantification and detection of small molecular weight metabolites, and these include amino acids and sugars, and basically all the biochemicals that we produce and organisms produce. It's a non-targeted approach, so you don't just measure one endpoint, you can measure a whole range of endpoints. And it's almost like a health checkup, so it gives you an idea of how the organism is responding in a more holistic way. Some of the work that Ollie's done has looked at the effect of pollutant exposure on non-vertebrate, non-model organisms, and he's shown that the effects, changes in metabolite profile occurred at concentrations below those where other endpoints were affected, like growth and reproduction. So metabolomics is a very sensitive tool to be used. And CAPM are using metabolomics to develop new techniques in non-model aquatic organisms, and specifically for Australian organisms as well. And what's the beauty about metabolomics is you don't need to have a genome sequenced. It's based on the chemistry of that particular metabolite. So what makes an amino acid in a human is the same as an amino acid in a chironomid. So you're sort of more looking at the chemical structure of the metabolites. So it's easily transferred between different organisms. So now I'll just quickly touch on what the difference between a targeted and untargeted approach is. So this is an example of a yeast cell, and this is an untargeted approach. So you can see all the different biochemical pathways that are occurring in that yeast cell. And an untargeted approach basically captures the majority of these types of molecules. So it gives you a broad screen of what's going on. Whereas a targeted approach is where you can focus in on either a particular class of metabolites or a particular pathway. It's more sensitive, and that's useful to use once you've done your broad screen and you see where effects are being observed, and then you can more delve into finding out what's actually causing the problem and what the mechanism is of a toxic effect. All right, that's the background. So now I'm going to talk about some of the work that I've been doing where we've used metabolomics. So the first case study is looking at invertebrates in freshwater environment and looking at the effect of metals, zinc and copper. So our organism of choice is the chironomid. The life cycle is well documented. It's got sediment-dwelling larval stages. So this is just a brief schematic of the life cycle of a chironomid for those who don't know. So you've got in chironomous species, you've got adult male and females. The females lay an egg mass in the water, and then a couple of days later, the larvae hatch and become associated with the sediment. They spend the majority of their life cycle in the sediment, and then they emerge from there as a pupa, and then they go into the air, and then they become adults again. So the life cycle just continues. So because they have sediment-dwelling larval stages, they're ideal for assessing waterways. They spend a lot of time in the sediment and also exposed to the pool water, and that's where a lot of the contamination finds itself. Chironomids are extensively used in research and standard toxicity tests, so they're really relevant for ecotoxicology. In the US and in Europe, there are OECD and US EPA guidelines for using chironomids. For us, they're relevant because they're one of the families that are recorded in the bioassessments. So they're already collected in Victorian waters. And for us, for the metabolomics, one of the main things is that they're easy to culture in the lab. So we can work out, we can manipulate their age, and we know what they've been previously exposed to. So for lab experiments, they're really useful to use. So the aim of the first experiment was to look at the effect of exposure to metals in chironomous teperi larvae. So that's a local species around Victoria in New South Wales. We wanted to identify zinc-specific metabolites through lab exposures. And then we wanted to validate the use of metabolomics in a field experiment using copper-spiked sediment. So the first experiment was to use 11-day-old larvae, so they were a third instar. And we used the third instar because their responses are quite conserved. Second instar is a bit more sensitive, but you get their response is very, very variable. So we exposed them for 24 hours to either water or 2 concentrations of zinc. And we worked out the concentration of zinc based on their LC50, which is a concentration that kills 50% of the population. So we used 10% and 1% of the LC50. So we knew that they would get a response, but we didn't want to kill them because we wanted to develop these sensitive responses to pollution. So we used 2 different types of analyses. We used an untargeted gas chromatography mass spectrometry approach, where we looked at around about 300 metabolites, and these were polar, so they're water soluble. And then we used a targeted approach, which was liquid chromatography mass spectrometry, where we just focused on amine containing compounds. They're compounds with an NH2 group. So this is just principal components analysis to look at the GCMS data. And what you can see is the zinc high, we're getting separation between them and the rest of the treatments. And we found 30 significant metabolites. And they were mainly sugars, amino acids, and we did get some unknowns. So we go back to a library and have a look and see what these metabolites are based on databases. And there's some that we couldn't actually identify. And one metabolite that looked really interesting was called cystathionine. So we delved in a little bit further to find out what it was involved in. And it's this pathway called the transsulfuration pathway. So basically it converts methionine through to cysteine and glutathione. And what was really interesting for us was that it's involved in the production of metallothioneins, which I said earlier was a biomarker for metal exposure. And because we were exposing the chironomids to metals, it made sense that we were seeing changes in this pathway. So cystathionine is crucial within this pathway. And because we looked at amine containing metabolites, all of these metabolites contain amine, so it was a suitable pathway for us to use the amine approach. So again, we got separation from the zinc high, and we saw significant metabolites. So this is what we found in this pathway. We saw a reduction in abundance of methionine, cystathionine, and a couple of other metabolites, but no difference between cysteine and glutathione. This could be because cysteine and glutathione come from lots of different biochemical pathways, so we just see what's the snapshot of the total pool of these metabolites, so we may not be seeing what's actually happening in this pathway. To do that, we'd need to do some isotope labelling studies where we can look at the flux of the different metabolites through this pathway. So just to summarise what we found here, we can use metabolomics to detect differences between zinc-exposed animals and controls. And there's a couple of biomarkers in the transsulfuration pathway that seem to respond to zinc. Right, so that's all well and good, it's great, we've got a lab study, but in the field, that's where we really want to be able to use this work. And as you can see, a lab study is nice and controlled, and in the field, there's a wide variety of different effects that the animals are exposed to, and we need to develop tools that are robust enough to see responses due to pollution that can respond over and above these environmental effects. So we use a field-based microcosm study. So this work was carried out by Katie Jeppy. She was a PhD student within CAPM at the time. So what Katie did was she had some microcosm containers, basically just plastic tubs, spiked with contaminated sediment with copper. She put these tubs out in the field site that's got high chironomid biodiversity. So the idea is you put the tubs out, you let the animals colonise, and after a few weeks you put stockings over the top so you collect the animals as they start to emerge. And then you can look at the diversity and abundance of these animals. One of the limitations of the microcosm study, though, is that sometimes you do get a patchy distribution of animals within the microcosms, and we don't know whether that's because the animals don't like the sediment because it's polluted, or whether they just don't like that particular position. So we need to understand a bit more about what's going on. So what Katie did to overcome that was 5 days before the end of the experiment, she put in 5-day-old Chironomus teperi larvae that she'd raised in the lab, which are here, into every single tub, and left them out for 5 days. And then when they finished the experiment, they were 10 days old, so a similar age to when we did our lab experiment. And she collected the teperi larvae, and we know that there's low abundance of teperi larvae at the field site. So anything that she found in the tubs were actually the larvae that she actually put in the tubs. And then she looked at the biodiversity and also did some metabolomics work on the teperi larvae. So this shows what happens to the diversity of the macroinvertebrates. You get increased copper concentration on the x-axis and abundance on the y-axis. So you can see as you get increased copper, you get reduction in biodiversity. And then Katie looked at the weights of the Chironomus teperi, and as you can see, an increased concentration, you get reduction in weight. All good. And then she looked at the transsulfuration pathway to see what happened here. And she found some significant results, which was really exciting for us. We'd never seen this before. So she got increases in some of the metabolites in this pathway. So we can show that in both copper and zinc, we're seeing this pathway changing in response to metals. And I'll just remind you of what we saw with zinc. So we're seeing different metabolites changing, and we're seeing different direction, whether we're getting increased or decrease in abundance of these metabolites. So this is really exciting for us. First time we could see this pathway responding in a field environment, and we could see differences between copper and zinc as well. So this pathway seems to be a good one for metal exposure. So just to summarise our work here. So Katie could detect changes in the macroinvertebrate community responses. She found that there was a reduction in growth, and she also found that there was changes in the transsulfuration pathway. So that graph that I showed that had the different levels of biological assessment where you can measure responses, Katie ticked the boxes for all of those, so you could link metabolomics to changes in the community. And so this is the first step in the field validation of using metabolomics to detect changes in response to metals. And Katie's published this data. All right. This is work that's part of an ARC linkage grant that's funded by Melbourne Water as our industry sponsor, and CSIRO and Metabolomics Australia as our collaborators. So we wanted to look at the effect of metals in an estuarine environment. So the questions I wanted to ask was, do all animals respond in the same way to the same chemicals? So when you go to a site and you want to collect some animals to look at what's going on, do you have to look at specific animals, or does all animals respond in the same way? They may be more sensitive or less sensitive, but are the same biochemical pathways changing? So it doesn't really matter if you get any animal. Somebody like me could go out and I don't know one thing from another. Whereas do you need a taxonomic expert to go and actually identify particular animals. So that would obviously increase the cost of your biomonitoring program. So do taxa respond in the same way? And are there chemical-specific metabolites that we can use as biomarkers? So to use metabolomics in a more diagnostic way. So I looked at 2 different animals. I looked at snail, Neisseria bacardi, and also the worm Simplicita equisetus. They were common and abundant in estuarine intertidal mudflats, and they were really easy to collect. So we collected as many as we needed in about 10 to 15 minutes. So again, in terms of biomonitoring, it will reduce your cost, your labour cost, if you want to collect these animals. And importantly, they were easy to maintain in the lab. We did collect a second snail species, because I wanted to compare the difference between 2 different snails, but unfortunately they weren't happy in the lab. So we wanted to use a species that was robust, and we know that the lab conditions weren't going to cause any stress. So we only used this one species. So again, we used water only. We exposed for 48 hours this time. We weren't sure whether we'd need to expose for more or less, so we did a pilot study and exposed animals for 24, 48 and 72 hours. And we found that at 48 hours that was a good time point to see changes. So we stayed with that one. We used high and low concentrations of copper and zinc, and again we used seawater controls, and we had no mortality throughout, so this is showing it's quite sensitive and not toxic. So we had one extraction, and we could use 2 different analytical platforms again. We used an untargeted approach to capture the global polar metabolites, and then we used the amine approach to look at the transsulfuration metabolites. So we processed the data using an internal standard normalization and a log transformation, and then we did statistical analysis. And then we did univariate stats to look for treatment differences. So for the GCMS, we detected 50 metabolites using a targeted matrix this time. And we had a range of different types of metabolites. And interestingly, the same metabolites were detected in both worms and snails. So all this basically shows is there was no separation between the treatment groups with the principal components analysis. So then we carried out univariate stats to then see if we could see treatment differences. And we could. So this is copper, and we looked at copper low and copper high. So what we can see is that stearic acid, which is a free fatty acid metabolite, is responding in the same way to both high and low concentrations of copper, which is really good. We're getting consistency between the treatments. Interestingly, we don't see the same metabolites in snails and worms exposed to the same contaminants. Furanose is a sugar, and GABA is a neurotransmitter, and fumaric acid and succinic acid are involved in energy metabolism. So it looks like different pathways may be being affected. And this is just a box plot to show the abundance of stearic acid with the different different treatments in snails. And then with zinc, this is polar metabolites, not the amine metabolites. Interestingly, then with worms, we saw succinic acid responding in both the low and the high, so that was really important for us to see that we're getting the same metabolite changing. And you can see here that the abundance changes with the different treatments. Okay, then we did LCMS, and we detected 50 different types of amine containing metabolites, and the same metabolites were detected in both species. And PCA shows something similar, that we can't really see any clear separation between the treatment groups. But when we did the univariate analyses, we could see some different metabolites coming out. And again, we see differences between snails and worms. We don't see anything consistent between snails and worms. And with this analysis, we don't see similar metabolites responding between the treatments either. And the same with zinc. But what we do see with zinc is that metabolites are changing from the same pathway. So epinephrine and tyrosine are involved in the same pathway, and they're responding in a similar way. And I just want to point out that tryptophan in the worm, we also detected a reduction in tryptophan using the different analytical techniques, so it seems we're picking up the same responses between two different platforms. So just to summarise this work, we didn't see any metabolites that changed consistently between the treatments or between the worms and the snails. So that first research question that we wanted to ask was, does it matter what animal you use? Yes, it does in this case. They were responding differently to the different treatments. With copper, the snails seemed to respond better to, and they responded in reduction in free fatty acids metabolites. And for zinc, worms tended to respond, and then we had changes in energy metabolism and amino acids. And we could see some species-specific metabolites that have potential for biomarkers of metal pollution, but that needs to be developed further. So the next steps for this work would be to repeat with species from the same family. I think I was being too optimistic to look at animals that were so taxonomically diverse. So we try with some more worm and snail species. And also it would be good to do a field study as well to see what happens after we're in field exposed animals. All right, so this is the last case study, and this is something completely different. So it's to look at fish health in Port Phillip Bay using the sand flathead. So this was a big collaborative project. It was a collaboration between CAPIM and Curtin University, with Martha Monick-Gagnon and Jared Baker as the lead. And there was other investigators involved in this work, so Barbara Novak from the University of Tasmania and Scott Wilson from Macquarie University. So just background. There's been concerns about fish stocks in Port Phillip Bay, for example, the sand flathead. And the last study was on fish health was carried out in 1999 by Monick-Gagnon, where she looked at sand flathead responses at different locations of Port Phillip Bay. And she found that there was, looking like there was some effects of different types of pollution. There's increasing anthropogenic pressures on the bay, increasing population, increasing urbanisation, and increased industrial discharge. There's quite a lot of work done to improve the discharge into Port Phillip Bay as well. So Monique found that sand flathead were exposed and taking up contaminants, and the main contaminants that she found in the 1999 study were petroleum hydrocarbons, metals, and agricultural chemicals. So we wanted to repeat the same study 16 years later to see whether we could see the same sort of thing. So the aims of this project were to evaluate sand flathead health via a suite of biomarkers. So we looked at physiological indicators, we looked at biomarkers, and we looked at pH metabolites. So pHs are quite easily metabolized. So you can't usually measure the parent compound, but you can measure the metabolite in the bile. We used metabolomics to look at perturbations in the liver, and also we analysed the muscle of the fish. And the reason why we used the muscle of the fish is that's what Monique did in 1999. So to be able to compare between the two studies, we used the same tissue type to look at what the concentration of chemicals were. So I'm just going to talk about these two things today. So these were our sampling sites. So Sorrento and St Leonard's were classed as our reference sites. Corio Bay, Hobson's Bay and Mordialloc were classed as our more contaminated sites. Recent study has shown that Hobson's Bay and Mordialloc had more elevated levels of hydrocarbons and metals compared to the rest of the bay. And historically, Corio Bay has had input from the oil refinery operations. And so we collected sand flathead from these sites, and we just focused on females, and they were 2 years old. So the analysis of the chemicals in the tissue, we could see that there was the concentrations of the organic contaminants were below the limits of detection. Arsenic, copper, mercury, selenium and zinc were all detected in the fish. And in terms of the bile pH metabolites, we found that there was differences in petrogenic metabolites between the sites, particularly Corio Bay and Hobson's Bay. So that shows that they were exposed to pHs at these sites. And Monique and Jared have both published this work recently. So for the biological results, we could see that there was increased LSI, which is the liver somatic index, in Sand Flathead at Mordialloc, Hobson's Bay and Cryobay. So that means that their livers, they had enlarged livers compared to the other sites. But there was no difference in the condition factor, so that's a length weight measure. And for metabolomics, we did find differences, and they were mainly in amino acids, energy metabolites, and free fatty acids. And I'll just show you some of those now. So the amino acids are on the top, and the colours are Sorrento is green, St Leonard's is blue, Cryobay is yellow, Mordialloc is purple, and Hobson's Bay is red. So as you can see, these 3 amino acids, we're getting increased concentration of those at the more contaminated sites. And the same thing with these, what we refer to as intermediates of central carbon metabolism or energy metabolism. And again, you can see that at the more contaminated sites, you've got increased abundance of these types of metabolites. And what we think is happening, based on what we've read, is that when you have metals and hydrocarbons present, change your energy metabolism requirements. So proteins get broken down and break down into the amino acid components to increase the amount of amino acids available for energy, which is what we're seeing here, and changes in energy metabolism just to cope with detoxification and oxidative stress. And again, for the free fatty acid metabolites, we looked at saturated fatty acids and polyunsaturated fatty acids. And again, we can see differences in abundance from the different sites. And it's thought that fatty acids are involved in energy production too, so it's more evidence that energy has been affected due to low levels of pollution that the fish is likely to have taken up from the contaminated sediment or also eating contaminated prey. So just to summarise this work, there was low-level chemical contamination, but we did see some biological differences. We saw changes in physiological indicators, and we saw some differences in metabolites at the contaminated sites. And so the biology, even though the chemistry didn't show that much difference, the biology did show differences due to maybe metals and hydrocarbons. And this could be a reflection of changes in energy requirements needed for detoxification and oxidative stress. So finally, I'd just like to sum up what I've shown you so far. So we've identified metabolites in lab for studies in freshwater and estuarine invertebrates, and a multiple platform approach is useful. What was interesting when we looked at the estuarine invertebrates, we didn't see any changes in that transsulfuration pathway which we saw in the freshwater, which I'm thinking may be related to the chemistry of the zinc and copper relating to the saline environment. I showed you about Katie's work on the semi-field experiment using a metabolomics approach, and also how metabolomics can be used in fish assessments to understand how fish respond to complex systems. So where to next? It'd be really useful to do some work with emerging contaminants, for example personal care products and vet medicines that are present in really low concentrations, but obviously you're going to have some sort of biological response. And metabolomics hopefully is sensitive enough to be able to detect that. It's really useful to know what happens in natural stresses, so temperature, salinity and nutrients, and how they impact pollution as well. And also mixtures. So we can see with both the freshwater and estuarine work that zinc and copper animals responded differently, so it'd be useful to see what happens when they're put together. Do we still see chemical specific responses, or does something else happen? And also responses over time and different life stages. I just want to touch on the different life stages. I had a student over from France last year, and she did some work on metabolomics in estuarine amphipods, Emmanuel Leber. So we're still analysing her data, but what she did was she looked at juvenile and adult amphipods, and you can clearly see on this dendrogram how the adults and juveniles are separating out, so we need to go through that data more. But you can clearly see that metabolomics can separate out different life stages. So I've got a lot of people to thank. I want to thank everybody for coming, people in CAPM, particularly Katie, Georgia and Emmanuel, who've really helped push this work along. And Alison O'Brien is the other postdoc who works on the ARC linkage project with me, and everybody at CAPM, staff and students past and present. This work couldn't have been done without Metabolomics Australia. We work really closely together at Bio21. They're involved in all aspects of the project, from experimental design through to analysis and interpretation. Curtin University, I want to thank Molly Gagnon and Jared Baker and Melbourne Water for their support of CAPM and also the ARC linkage. And that's Rhys Coleman, Melita Stevens and Judy Blackbeard. I really want to thank SETAC Australasia for funding me to travel around Australia and talk about environmental metabolomics. Thank you very much. And I want to thank the Australian Research Council for funding our ARC linkage project, and Melbourne Water and CSIRO for being collaborators on that project. And thank you all very much.
Thank you, Sarah. Wow, what an interesting talk. Any questions?
Sarah, I was just wondering, in terms of the actual testing, so looking at the water-soluble metabolites and amines, I'm just wondering, what about like the non-water-soluble and specifically lipid and its relationship to organic contaminants in terms of that binding and metabolism pathway?
Yeah, we can definitely, you can measure the lipids as well. It's just a different extraction technique. So we just focused on the water-soluble ones. But yes, you can definitely do lots of different types of analyses.
I'm just wondering because like interesting results in terms of the water-soluble, and you can see in terms of the amino acids and and the protein synthesis and how that relates to metal metabolism, biometabolism, but also just whether you could have similar bioindicators in terms of
definitely.
In terms of using perhaps the lipid as a metabolism pathway.
Definitely. Yeah, because the organics are likely to bind to the lipids within the cells and things. So yeah, that's definitely something we're thinking about.
Thank you so much for the presentation. My research is particularly focused with the amino acids. So I have a question with the last case study. You showed that there was an increase in some particular amino acid level according to the pollution sites.
Yeah, well, the polluted sites had higher levels of amino acids compared to the...
Please explain why it was seen, like why the polluted sites, the organisms have higher level of amino acids.
We think it's because these animals need to break down proteins to release the amino acids to be involved in energy metabolism. So it's due to the animal's response to metals and also other contaminants to provide extra energy for them to be able to cope with contamination, because they have to respond to that contamination by producing more proteins and just using a lot more energy. So it just gives more energy available for these animals to be able to cope with the pollution once it's come up into them. Does that make sense?
Yeah.
Okay.
Good question. Anyone?
This is what I've been dreading.
No, I don't want to ask you. So you mentioned your Port Phillip Bay survey, repeating work that had been done. Can you tell me, is Port Phillip Bay better than it was?
Yes, much better. Yes.
Oh, that's good. That's a nice easy one.
Yeah, well, certainly in the tissue there was the organic contaminants were below limits of detection, and the metals were low as well. So when we compared what we found in the tissues of the flathead from Port Phillip Bay to one from clean sites, you would say that there would be really no difference.
Oh.
Which is good.
What do you relate that to, Sarah? Like in terms of the fact that, like I immediately think of the 30 million cubic metres of sand that was dredged in the bay and the channels, and these are channel rats essentially.
Yeah.
Whether that might be the fact that it's a cleaner system post dredging.
I think, but dredging could also mobilise some of the contaminants too, and people have shown that hasn't happened. I think it's improved effluent treatment as well, so there's a lot better treatment of water going into Port Phillip Bay too. And a big push for, after the Port Phillip Bay study, I think there's been a big push for cleaning up the bay.
You mentioned that this could be used in conjunction with biomonitoring tools. How do you envisage that working?
I think at the moment, I think what we would do would be to use it with the standard toxicity tests. So we would use animals that we raise in the lab, expose them into the contaminated sediment that have been collected, and then take the animals and do metabolomic analysis on that. I think we still need to do a little bit more work in refining our processes for field collected animals. We need to look at different age effects and things like that, but I think if we can use it in conjunction with lab experiments where we bring the sediment in, I think that would be really useful as well.
Sarah, I remember 10 years ago, Sara had issues with, in terms of the species and being able to keep them healthy in the lab.
Mm-hmm.
And yet when you're bringing species in, in terms of the animal husbandry around that, is that,
That's right.
you see that as a bit of a challenge?
It is, yeah, that is the challenge. That's why we try and have the animals either, if we can't raise them in the lab, we keep them and make sure that they're happy to be maintained in the lab for a few weeks to acclimate to our lab conditions before we start then doing some work, because metabolomics is really sensitive. Metabolites can change within seconds, so we really need them to be happy and acclimated into the lab.
Just going on from that, because in the lab you might alter their diet to what would be in a natural form, which could also possibly alter the fatty acid composition and stuff like that. So how do you envisage linking the results that we see in the lab to possible field effects if they've got the altered diet?
That's a really, really good question. I think that's something else that we'd have to look into further. But I guess you just compare what you see in the lab. You'll have your control animals as well. So you'd look at how the controls are responding and incorporate those changes as well. But it's something that we do need to think about. That's a really good question.
I've got a question. So, I was just curious, you mentioned veterinary medicines and pharmaceuticals at the end. That's something we're interested in at RMIT as well. Have you got any, well, my group at least, do you have any is that something that Marlborough Water said we're interested in, or is that just general environmental?
Just general environmental, I think.
No.
Can I kind of follow on to
Yeah, definitely.
So, kind of relates to that. So in the snails and the worms, you saw differences in which metabolites were up and down regulated following the exposure. So my question is really, is much known about the metabolomes of those animals? And then the way this then relates to what Ollie's just asked is, can you look at kind of existing libraries of metabolomes of other animals or something to identify what types of pathways might be like target pathways for, you know, like say going with some knowledge of what might be affected by?
I think so. There's not a huge amount known for those particular animals, so it is sort of a bit in the dark at the moment. But certainly you can look at related types of species as well. Yeah. And then you sort of try and understand where you're going to be seeing effects, and then you can sort of do a bit more focused metabolomics.
So you can kind of work backwards.
Yeah.
I'll just add on to that. So we've done just a rough scan of different macroinvertebrate taxa on the amino acid composition. Okay. And we're finding more, aren't we, Manisha, that it's based on biology, rather than like differences in diet or anything. So it's based on their genetics.
Oh, yes.
Yeah. And there's work being done in the UK and America where they're doing just that. They're trying to look at the genomic profile and they can predict what species are going to be affected by particular chemicals based on their genome profile due to their evolutionary history and things.
Any more questions?
Yes. So a quick one. In terms of the analysis, chemical analysis that you do, is using GCMS for the non-targeted, to find the non-targeted metabolites that you're looking for. Is like GCMS the only way to do that, or are there other options?
There are other options. There's another method, and I don't know whether you use it, called HILIC. That's a liquid chromatography method. So I think you can look at 2,000 different types of metabolites. But then what you would do, you wouldn't identify all 2,000. You would compare treated and untreated, look at where you've got differences, and then start to identify those. Yeah. But yes, you can use the liquid chromatography.
And then
And yeah, and then of course, yes, sorry.
Any more questions? If not, thanks again, Sarah, for a wonderful talk.
Thank you.